Hi, my name is Megan Nickerson, and I study ecosystem genomics at the UREN lab in the Bio5 Institute. So I'm just going to go into a little bit of our lab focus and background before I dive into my specific UBERT project. Just so as an introduction, our lab studies the biodiversity, biogeography, evolutionary origins of plant-associated microorganisms. So we're interested in characterizing the biotic and abiotic factors influencing microbial communities, how these communities impact their ecosystem function, and the evolutionary dynamics in the context of these plant-associated microorganisms compared to pathogens. So how aren't these pathogens? So the specific plant-associated microorganisms that we study are called fungal endophytes. So they live within plant tissues, specifically the ones we study live within leaves of plants. And they're unlike pathogens in that they don't provide any visible symptoms of infection. So you wouldn't necessarily know they were infected until you tried to culture from them. And then the relationship to their host is very complex. There have been some that have been shown to be beneficial to their host, but basically the most that we know is that they're filling the niche of a pathogen. So like a pathogen could be living in the plant, but the endophyte is blocking any sorts of pathogens from living there. And then it could be even more complicated than that, but we're still trying to figure that out. So we study these microbial communities in two ways. We have our traditional culture-based microbiology assays where we take from the leaf and culture onto growth media within the lab. So above that, number one, I have some images of fungal endophytes that have been isolated, and you can kind of see the morphological diversity that we observe. There's lots of different structures, like whether they're fluffy or if there's like spots or if it has mycelia, that kind of thing. And then number two, we use next generation omics tools. So basically there's something called culture bias in microbiology, which is a large hurdle because there are lots of microbes out in the world that don't necessarily grow on our media. They grow in the wild, but they can't necessarily grow by themselves on our media. So by using these tools, we can look at the DNA, RNA, and proteins in a community and identify who is living there a little bit more accurately. So that brings me to my UBERT project. So I focused on how leaf chemistry influences the microbial communities that we see in oak leaves. So plants produce something called secondary metabolites, which are compounds that aren't involved in primary metabolism. So that means that they are just accomplishing other things for the plants besides just staying alive. So specific secondary metabolites that I study are polyphenolic compounds, which have been shown to do a lot of different things, but they deter herbivores, they defend against pathogens, and they do things like defend against UV. So I was interested in studying how these polyphenolic compounds influence these fungal endophytes, and that hasn't really been studied in depth before. And so I wanted to determine what role they play, both their type and concentration, in the abundance and community composition of their foliar endophytes. So in order to do this, we collected oaks from throughout North America. And so this is a phylogenetic tree of the different oak species that we collected from. We tried to get a wide variety of different oak species. And then the little colored boxes, the pink, yellow, or blue, tells you where in the world we took it from and what kind of climate. You can kind of get an idea of the climate that they're living in. And then the other colored boxes tell us if the leaves are evergreen, brevideciduous, or deciduous. So once we have our leaves and we know what kind of environment we're dealing with, we would grind up our leaves and they would go into three different categories. One category they would go into is measuring the total polyphenolic content of the leaf. And then two, we would grind it up and do a DNA extraction to see who's living there without culturing. And then the third one is we would put it into a metabolomics assay, which would use mass spectrometry to determine the specific chemicals. So we might know the total concentration, but we want to know what exactly was in there. And then currently we are growing endophytes on media that contains polyphenolic compounds at different concentrations and type. That way we can see in vitro if there are any trends that might carry over into the actual living leaves. So just in summary, our lab studies the intricacies of plant-associated microorganisms through culturing and next-generation omics to better understand their roles in ecosystem structure and function. And then these are just our lab members here. And then these are my works cited. Thank you.